Welcome to 843 TV. I'm your host, Carrie Manning from Heritage Dental Spa and Salon.
And I'm Will Ware. I'm with WHHI-TV. Today we're coming to you from USCB in the Bluffton campus, which brings us to our first guests, Dr. Kimberly Ritchie and Dr. Edward D'Antonio, both professors in the biology department.
And then we also have 3 students who are currently enrolled here in the biology program at USCB. We have Daniel Conrad, James Lanier, and Scott Green. So make sure that you come back and join us for more 843 TV.
Where communities come to speak.
(843 TV), where Bluffton comes to speak.
Where Spring Island comes to speak.
Where Hilton Head Island comes to speak.
Where Beaufort comes to speak.
(843 TV),
where communities come to speak. Welcome to 843 TV. Today we're at USCB and we're joined by Dr. Kimberly Ritchie and Dr. Edward D'Antonio. Thanks for joining us.
Hi, thanks for having us.
My pleasure.
Absolutely. And so you're both members of the Natural Sciences Department here at USCB, is that right?
Yes.
Okay, and Dr. Ritchie, you're Associate Professor of Genetics and Prokaryotic Cell Biology. And that is a mouthful. Can you tell us a little bit about what that means?
Well, my areas of expertise are microbiology and cell biology, microbial interactions with their hosts. So that's where that came from. And my PhD is actually in genetics. So. Okay,
that's cool. And there's been a real kind of popular interest in genetics here with all of the genetic kits and 23andMe and all those things becoming more popular in mainstream America nowadays, hasn't there?
Yeah, there certainly has.
So what got you interested in genetics? And
well, my main area of expertise is actually coral marine biology, so coral reef biology, and I studied the interactions between symbiotic bacteria and coral reefs. And corals. Cool. So everybody has symbiotic microbes, and that's what I'm mainly interested in. So I wanted to get a background in genetics so that I could apply that to marine systems, because that wasn't being done at the time that I was getting my PhD. So that's my idea behind that. Okay, and so where did
your educational history, where can you take us through that a little bit?
Sure, I actually got my undergraduate at USC Aiken, and that's where I started research on coral microbe interactions, and my PhD in genetics is from the University of North Carolina, Chapel Hill. Okay. And then I went on to do postdoc at the Smithsonian in Panama and Scripps Institution of Oceanography, looking at host microbe interactions with coral diseases and then beneficial associations of bacteria and corals.
Okay. And so what brought you here to Beaufort and USCB?
Well, I then worked at Mote Marine Laboratory for 13 years in Sarasota doing strictly research, and I'm from South Carolina, so I wanted to come back. That's always been the plan, come back to South Carolina, and I love the idea of being part of a school. USC Beaufort, such a great area with such a growth trajectory, so I'm excited to be a part of that.
And right in the middle of the Port Royal Sound area, which is abound with marine life, right?
That's true, and a lot of it is very understudied, so it's a wonderful system for anyone that works on microbial anything interactions. So I have my pick of things to study, and it's such a wonderful, beautiful area.
Great, we're glad to have you.
Oh, wonderful.
And Dr. D'Antonio, you're the Associate Professor of Biochemistry and Structural Biology. So what's your educational background and research?
Okay, well, I study neglected tropical diseases, and one disease in particular is called Chagas Disease. And that's basically my main research interest, is to do drug discovery, and we're doing structural biology and designing drugs by using this technique to bind to enzymes with very strong inhibitors.
Well, I know this is not on your page, but with the onset of antibiotics shortages and all that stuff, is that connected to what you're doing, or is that separate?
Well, we actually don't use antibiotics. We actually use small molecule inhibitors. And these inhibitors go after specific targets that are found in the organism. So it is a little bit different in terms of the strategy that we're using. So
I think that stuff's fascinating though. So what brought you here to USCB?
Well, I applied to colleges on the East Coast, and I'm actually from Philadelphia. And when I got the interview to come down to USCB, I was like, okay, this is a great place because I have sort of like a Charleston state of mind, and I also have a North Florida state of mind. And I said, on
Meet
the
you in the
weekends,
middle.
yeah, the geographics are great around here, and we also live 20 minutes from the beach.
Not a bad deal, is it?
Yeah, so geographically it was a great idea, but in terms of academics, this was a nice place to start, as a research scientist, and that's sort of what I wanted to do. And we have a really good library. We could pretty much get any kind of article that we want. It makes a good place to write research grants. We have a lot of internal funding. And when I was hired, they offered me a pretty big research package for grants. I was given a South Carolina INBRE grant, which was about $120,000 over a course of 3 years to get my research on the road.
That has to be helpful.
Oh, absolutely, yeah. So I'm very thankful for that, and and when they told me that's what I was going to have coming here, I said, this is going to be a great place to start to be a research scientist, and it's a university where we could work with undergraduates.
Right.
And my goal is to get an undergraduate to the next level, as opposed to working at a Research One institution where it's a different philosophy. So you get to work with very nice students. You get to be a professor, teach the general courses, but you could also, have that fun where you're doing your research.
Well, and I think it's an interesting opportunity for you all working with undergrads too. They're eager and they're yearning for so much information, and I think they're trying to find their specific focus too. So that
Yes.
is a different perspective.
Undergraduates come with what I call a young energy, and there's this motivation that comes with it because it's brand new to them, and it's a lot of fun.
I always loved to see research. Then somehow ended up doing hair. I took a hard right.
Well, Dr. Ritchie, Dr. D'Antonio, thank you very much for joining us today.
Oh, it's our pleasure.
All right, thanks.
And stick around for another segment of 843 TV. Welcome back to 843 TV. We're joined again by Dr. Kimberly Ritchie and Daniel Conrad, who's a freshman biology major here at USCB. Thanks for joining us.
It's our pleasure.
Dr. Ritchie, we're talking a little bit off camera about the OSearch vessel and some of the cool research you were doing with sharks and other marine life. Can you tell us a little bit about that vessel and that program?
Yeah, so the OSearch program is wonderful. So I talked about working on host microbe symbiosis, and we're starting to discover that sharks have this wound healing capability. They're able to heal their wounds very quickly. In their part of their ordinary lifestyle, they receive these traumatic injuries due to the way they reproduce and aspects. So we're interested in figuring out how they heal their wounds so quickly, and we're starting to see that there may be a microbial component. So I've been working for the past 4 years on stingray microbes that produce antibiotics for the host, and this is my part of the project on OSearch. So OSearch itself is this wonderful way to maximize on science because they very carefully capture these great white sharks and bring them up on a platform, and in a very controlled manner, within 15 minutes, we collect enough samples and data for 50 scientists to do research on various different aspects of sharks. So this has been a wonderful program, maximizing on information that we didn't know about white sharks, including where they go, because the final thing they do is tag them, and now we can follow, everybody can follow in real time where these sharks go now.
Oh, that's so cool.
I think that's a really neat program too, because even my girls, they talk about that at school. They follow those programs, and then it all goes viral very quickly on social media whenever Miss Mary comes and visits really close. But it's neat because I think it exposes a lot of people who wouldn't normally either be interested or have a clue knowledge is always...
Right, and now we're learning that they do come close, and they probably always have, and they haven't been a problem. So.
Right. Yeah, that's interesting. And in our last segment, Dr. D'Antonio was talking about some of the youthful energy that undergraduate students bring to this research. Dr. Ritchie, you've involved freshmen in your research, correct?
Yes.
How is this, in your view, how do freshmen or undergraduate students provide you an advantage in your research?
Well, I came from an organization that was strictly research, and we had a really strong internship program that was all undergraduate, and I've mentored over 65 undergraduate students, and some of these have now have faculty positions at major universities, and so I really love mentoring students. But the advantage of mentoring students at USC Beaufort is that I get to keep them for longer than 3 months. So one of the first courses I taught here last semester was general biology for majors. So I scooped myself up some ambitious freshmen so that I could help train them over the years. So that's a little bit different because they're brand new students. But the enthusiasm, and this is one of my most enthusiastic students, is infectious, and I really love training students.
That's very cool.
So speaking of you having many years ahead, that's exciting. So how do you feel like it helps you starting research earlier than what most students do?
I think starting so early in your undergraduate career is important because it's not only being proactive with the professor, you're learning these methods that they teach you, and each time you do them, you're perfecting them. So it's being proactive because there's nothing wrong with bringing a junior or senior on board. But when you do that, they're going to leave in 1 or 2 years. So I potentially have 3 more years with Dr. Ritchie, and I think that's really important because, like I said, we're perfecting these methods that we're learning each time we do them.
That sounds incredibly exciting. So what type of research are you helping with in class?
In the lab, we're looking at beneficial microbes and possible beneficial antibiotic-producing bacteria. And we're really fascinated with sharks and rays and their innate immunity. And we're looking at the epidermal surfaces of those marine organisms and taking samples and then doing all types of cool things in the lab with them.
And then how do you feel like that helps you when you go back to just traditional classwork?
You know, it really does help because I feel like having that hands-on experience when I go back to a lecture or I'm reading the textbook, I'm like, Dr. Ritchie had already touched on this. And it just really helps me put things into perspective.
I've always felt like that. I think anytime you get your hands on something and then whenever you cross those over, it makes it so much easier. Well, Dr. Ritchie, can you tell us about the other students that you have helping you in the lab? Like, what are their goals?
Yeah, I have all freshmen, and they have various different goals. Most of them want to do something in marine biology, and they're not entirely clear on that, so I really like that. And 2 of them like to fish. Daniel loves to fish.
Love it.
So that works out well because they're going to be collecting some fish, so it works out well that they're going to be doing that. I've got Aubrey Denton, Danielle Young, McKaylee Ford, and Olivia Whipple. These are all very enthusiastic young students that are excited to learn something. So what they're doing right now is just learning all of the techniques in my lab that include antibiotic screening and bacterial identification and genetic DNA sequencing for identification of organisms, and then they'll kind of spin off in their own projects once they get the general training.
Yeah, what a place to do this type of work. I mean, this is at USCB. You've got access to just a wide variety of marine life, and I think this is very cool. So thank you both for joining us and sharing with us a little bit about what you do.
All right, it's our pleasure.
And stick around for one more segment of 843 TV.
Welcome back. We're joined again with Dr. D'Antonio, and now we're being joined by Scott Green and James Lanier, both are pre-med majors here at USCB. So we'll start with you, Scott. Can you tell us a little bit about Chagas disease and how it's transmitted?
Yes, ma'am. Chagas disease, it's one of the world's neglected diseases. It's actually a neglected tropical disease, and currently it affects about 8 million people worldwide. mainly in impoverished nations in Central and South America where, medical treatment is either very scarce or, they don't have the money for it. And it's spread by an insect vector. It's called a triatomine insect. It's a kissing bug. And what happens is the kissing bug bites a person, and in the process of them biting a person, some of their feces actually get into the bite wound, and the parasite, Trypanosoma cruzi, is what is inside of their feces, and that's what gets into your body, and it starts to get into your bloodstream. It starts hijacking cells and feeding off of blood glucose. So basically, in the acute stage, some of the symptoms you can see are, localized swelling. You can get stinging pains in your fingertips. You can have diarrhea, and as you move into the more chronic stages, you end up getting, as these things start to build colonies in the soft tissue around your organs, you can end up with cardiomyopathy, enlargement of the heart. You can end up with a megacolon. You can end up with a megaesophagus. Very uncomfortable. And basically, it affects a person based on their overall health.
Which generally people in impoverished areas don't have the best health to start with.
Exactly. So, and then if you have someone who's really young, who doesn't really have the developed immune system yet, or you have someone who's older, who maybe have a more weakened immune system. And so basically in the last, since the early 1970s, there hasn't really been a whole lot of progress on this. There was 2 drugs that were developed in the early 1970s. They were called benznidazole and nifurtimox. And basically, they're both called prodrugs. And so basically what they do is they go into the body as an inert drug, and they pass through cells. Once the drug actually finds the cell that contains the parasite, it activates. And it kills the cell, but it also becomes toxic to the body. And so...
Problem.
Yeah, it is. The idea is to make a medication where, you're going in and you're harming the bad cells and you're leaving the good ones alone.
Stuff. Yeah.
So there's a real need right now to develop new drugs that are going to be selective in that way, to go in and, take care of all the bad stuff and leave all the good stuff alone, so that the person's only symptom is, they feel better.
Right.
So basically what's happened is Dr. D'Antonio has identified a metabolic pathway that targets this parasite and kills them. And by a process that we call structure-based drug design, we can basically take the first step in this metabolic pathway, and if we can kill the parasite right there, then it stops the cycle.
That's impressive.
So it's really interesting, actually, yeah.
That's amazing. I love that kind of stuff. But well, you mentioned the structure-based design with Dr. D'Antonio. What is involved in synthesizing and testing all these new compounds? I mean, this sounds incredible. It doesn't sound. It is incredibly detailed, very intense information here right at USCB, which I think is a very valid point to make.
Well, structure-based drug design, this is just a technique of drug discovery that actually falls under an umbrella of another technique called X-ray crystallography. And our lab uses this technique called X-ray crystallography, in which I have students purify proteins to almost 100% pure. And then what they're going to do is they're going to grow a protein crystal. So from solution to the solid state, you can observe a crystal of the protein under a microscope. Now this protein crystal is going to be that of the enzyme that's found inside of Trypanosoma cruzi. That's going to be called our drug target. Now if we can get that crystal grown, we send the crystal out to a national laboratory in Chicago. It's called the Argonne National Lab. Also, there's the Advanced Light Source. And I have a collaborator there. She mounts our crystal in front of a beam line. And with a click of a mouse on her computer, she opens the shutter of the beam line, and X-rays fly through and they hit the crystal, and then you get an X-ray diffraction. We take the diffraction and we can take that data and build a 3-dimensional structure at the molecular level of our protein. By having the vision of the 3-dimensional structure of our enzyme, we can then use structure-based drug design, where I know what the inside of the enzyme looks like. I know the interior. There's a microenvironment. enzymes catalyze reactions. So what we try to do, we want to get an inhibitor that we could design that's going to basically sit in there and just make contact. And if we can do that, the metabolic pathway of glycolysis would be, the flux would be constricted, thereby leading to apoptosis of the Trypanosoma cruzi.
That's kind of amazing. And I'm sure there are some people that are like, what did he just say?
Well, now, James, you're one of the students who's helping Dr. D'Antonio with this research, you and Scott. Can you tell us what some of the compounds you're currently working on and how you attempt to synthesize and purify them?
Well, instead of like just kind of spewing off a bunch of names that nobody knows, I'll tell you kind of like the process. Okay. The overall goal is to create a glucosamine analog inhibitor. And what we've done to create this is we went and we bought 7 commercially available acyl chlorides. And what we're doing is we're taking our glucose molecule and we're binding it to our glucokinase. Now, what that pretty much means is we're taking our glucosamine molecule and we're reacting it. We're going to react it with one of those 7 acyl chlorides that we bought. So then the hope is that it's going to strongly bind to the enzyme.
Yes.
And so that's pretty much the synthesization process. Okay. Now, for purification, once we synthesize it, we have like a bunch of leftover, like there's the things that we use to synthesize it. So there's all kinds of byproduct. So we have to purify it. We have to get it down to the nitty gritty. Because if we have all this extra stuff, that's not what we want. That's not our compound. Right. Now, once we get it, we have to put it through our high performance liquid chromatography machine. Ours is god awful old. It is so old, but I mean it works. It works. It just doesn't have all the bells and whistles, but it does its job. Now, whenever we put it through this machine, we get a little graph and we get a peak. Whenever we see that peak around maybe 20-25 minutes of putting it through our machine, it's a rush. It really. I mean, for me, I don't know about it. If someone were to look at this graph, they're like, "Oh, that's just like something going up." No, for me, that's huge. That's our compound coming out. So it's really exciting. One time, I ran out and I knocked on Dr. D's door. I was like, "Man, we just got this awesome compound." You have no idea. But anyways,
hero peak.
Yeah, we call it a hero peak. It was great.
Yeah,
but anyways,
that was a big one.
Yeah.
Well, I think the moral of the story is that you guys are making amazing research. And I think a lot of people may underestimate our little USCB campus, but I think it is proof that it is growing.
Yeah, absolutely.
And that if you need more information, please visit their website. There are many programs and it's growing quickly. So thank you all for joining us, and make sure that you join us next time on 843 TV.
Where communities come to speak.